lecture 3 of enzymes, and we're going to start this lecture with a question that says, how do we know that the rate of an enzyme becomes constant? So the rate of an enzyme becomes constant when their number of substrate are greater than the number of an active site. The number of substrate is greater than the number of active site. We're going to discuss it with the help of a diagram that says, for example, this is an enzyme, and this right over here is the active site of an enzyme where the substrates are going to be attached. And these are the substrates. Since we know substrates are very specific in nature and that does not allow any enzyme active site to be attached to them, thus the specific amount of the active sites are available for substrate to be attached. For example, there are 10 active sites per se, and the substrates are 100, or the active sites are 100 and the substrates are, let's say, 10,000. Thus in turn we say that when the number of substrate is greater than the number of active site, what does the substrate do? That they starts to line up in front of the active site in order to be attached to it. Then we say when the rate of a limiting factor becomes greater, we say that the rate becomes constant of an enzyme and no further substrate can be attached to an active site of a certain enzyme. Now, what else we're going to discuss in this today's topic is factors of an enzymatic activity that affect basically its reaction, which consists of 4 of them. That says temperature, pH, enzyme concentration, and the substrate concentration. We're going to discuss each of them in detail. Let's start with the temperature first, how the temperature affects the activity of an enzyme in the reaction. Let's have a look at this graph. This graph shows us the temperature movement and how it works in an enzymatic activity. Let's name it at 0 degree, 10 degree, 20 degree, and 25 degree. We say that it works best at 20-degree temperature, and when the temperature rises above the 20-degree, it denatures, or the temperature is less than 20-degree, it does not work very efficiently. The temperature here after the 20 degree shows us the decline in the enzymatic activity, as that the enzyme is not working very efficiently. Whereas the increase in the temperature shows us that for every 10-degree rise in the temperature, the enzymatic activity doubles. Doubles by mean we say it becomes 2x of its original activity in order to be performing very well. The temperature plays a very important role in the enzymatic activity. If the temperature increases, then its optimum temperature is the temperature that is basically at which the enzyme works at its best. It becomes denature, and if it lowers than its optimum temperature, it does not work very efficiently. Next thing that we are going to discuss is the progress of an enzyme in a reaction. How can we determine what is the progress of an enzyme in a reaction? We can be describing this by the help of these two steps that says either we can measure the concentration of a reactant or we can measure the concentration of a product. By the help of these two, we can determine whether the enzyme is working very efficiently or not. So, for example, let's take an example. Let's say is reactant is being converted into the product. We'll be explaining these 2 points that say if the amount of reactant is decreasing and after the reaction is proceeded, the product is increased, then we can say the reaction is efficiently working and the enzyme is doing its best. Or the other way around, we can say if the amount of product has been increased efficiently and the reactant has decreased, either it's a reversible reaction or a irreversible reaction. So we can say that the enzyme is working at its best. These are the 2 points by the help of which we can determine this. Next point we have is the pH. We'll be going to discuss pH in detail. That says contains an R group of amino acid with proper charges. And another point says loose activity in low pH or high pH. As we've talked about it in the temperature, there is an optimum temperature. Similarly, there is an optimum pH at which certain enzymes work. There is a different optimum temperature or optimum pH for a respective enzyme. It says loose activity in low pH or high pH as tertiary structure is disrupted. Alright, I'll explain you by the help of a graph that says we'll be discussing 3 of them. three enzymes in order to understand. That says, for example, let's take, let me draw it. Okay. Let's say the first one that we have is pepsin. All right. Other one, let's take salivary amylase. And let's say our third one is alkaline phosphate. Since we have taken these three enzymes for the example, we are going to give them some of the temperature as pepsin works best at 1.5. Let's take it 3, let's take it as 4 and 4 and 5. Okay. So, since we have talked about it that all the enzymes work best at different optimum temperature, thus salivary amylase cannot work best at 1.5. Similarly, pepsin cannot work best at 2.5. Or alkaline phosphate cannot work best at 2.5. Neither can the salivary amylase can work best at 3.5. This basically helps us understand that for every enzyme there is a different optimum pH, and it requires that pH in order to give the best of results. So we need to provide that those conditions and that temperature and that pH to an enzyme in order to form a product of our needs. The last thing that we're going to discuss in this topic is the categories of an enzyme. As we know, enzymes are basically classified in the two categories, that is extracellular enzyme and the other one is the intracellular enzyme. These tools are very different from each other, as the name itself helps us understand it. Extracellular enzyme means that it is mostly present outside the certain organ or outside the body, whereas intracellular are the ones that are present inside the cytoplasm, inside the body, inside the cell. It can also be very helpful when we are going to discuss about some of the examples. The example of extracellular enzyme is pepsin. We are going to come back to this on how pepsin is an extracellular enzyme, whereas intracellular enzyme contains all the enzymes that are involved in the aerobic respiration. This is pretty simple to understand, but when it comes to pepsin, let's say that pepsin is, this is a stomach, and pepsin is present right at the wall of stomach. And from this wall, pepsin is being released into the stomach in order to digest all the yum food that we all have swallowed. By this term, we can really understand that since the pepsin is present outside the stomach, thus it is termed as the extracellular enzyme. One thing that we need to discuss before we end the lecture is that how do we know, or how we'll get to know in how the enzyme is not involved in a reaction, and what would be the consequences if the enzyme is not involved. One of them is that the reaction would be very slow. In any reaction in which the enzyme is not included, it becomes very slow. The product form that must be singular. There are very less chances that if there is no presence of enzyme in a reaction, the product must be singular. And also that the activation energy of an enzyme would be very high, thus it would require much amount of energy in order to kickstart a reaction and form a desirable product. It is very important to use enzyme in a reaction, as the enzymes are biological catalysts. As we have learned in throughout the lecture, they are the biological catalysts that basically speeds up the chemical reaction without being involved in it. Here the enzyme lecture comes to an end. I hope this has been very helpful for all of you.